In this podcast, I'll be talking about the second part of photosynthesis, the light-independent reactions, or the Calvin cycle. First, a little review again of photosynthesis. The overall equation that we're working with is photosynthesis requires water and carbon dioxide and, of course, sunlight. And then the process makes glucose as well as oxygen gas. We talked about in the light reactions how water is split because of the action of sunlight, and as a result the oxygens in water form oxygen gas. We have yet to talk though about where the CO2 is used and how the glucose is made. So we'll be getting to those today in the light-independent reaction. A review again of where we are in the cell. Okay. Within any organism that undergoes photosynthesis, we're going to have many little structures called chloroplasts. One of those chloroplasts has many different stacks of what we call grana. Each of those grana are made of thylakoids. So one of these little pancakes is a thylakoid. Each thylakoid has a membrane and a space within it. Around the thylakoids, the filling of the chloroplast is called the stroma, and that's going to be important for the processes that we talk about today. The light reactions, recall, happened along the thylakoid membrane. Okay, one more slide for review. The location, as I said, of the light reactions that we talked about was that thylakoid membrane. I'll abbreviate this time. And the purpose was to make some ATP and NADPH, which, as I said, are going to be used in these light-independent reactions, or the Calvin cycle, that we're talking about today. The location of the Calvin cycle is the stroma, that filling of the chloroplast, and the purpose, our goal for today, is to make glucose. All right then, let's go to the Calvin cycle and see how this glucose is made. So let's look at this little diagram up here on top first. Move this out of the way. All right, this is the one we want to look at first. Here is your thylakoid, okay, within the chloroplast. Set of thylakoids, actually, the grana, and in there is where those light reactions occur. This little yellow part in the middle is supposed to symbolize the ATP and the NADPH that's made in the light reactions and moves on to the Calvin cycle. Calvin cycle, which we're talking about today, is right here in the stroma of the chloroplast. Okay, so that's kind of our big picture. Now we're going to zoom in on that Calvin cycle and take a look at what happens. Is going to look kind of like the Krebs cycle from cellular respiration, or the citric acid cycle. We kind of have the same idea here in that we're having different carbon-containing compounds that get changed from one form to another, and they are recycled to keep the cycle going. So in this example, it's going to be important to keep track of the carbons as well, just like we did in the citric acid cycle. Another thing we need to keep track of is how many molecules we're actually dealing with. So I'm going to lead you through this once, like I usually do. I want you to listen first, and then we'll talk about how you might want to diagram this in your notebook. All right, here's where we use our CO2. In the stroma, we've got a 5-carbon molecule. We're going to shorthand call that RUBP. Okay, this 5-carbon molecule has 2 phosphates on either end of it. That's the yellow. The carbon is the gray. Hopefully you figured that out. And there are, in this cycle, when we talk about it when we're all done, we're going to be dealing with 5 molecules of RUBP. Okay, so we have 5 molecules of RUBP. No, 3, sorry, back up. I gotta keep track of my numbers. 3 molecules of RUBP. So, you're not diagramming this yet, which is good, because I already made my first mistake. So we've got RUBP, 5 carbons, that's where I was getting confused, 2 phosphates, and then 3 total molecules of RUBP is what we're going to be working with. To those 3 molecules of RUBP, we're going to add 3 molecules of CO2. There's our carbon dioxide. Now where does the carbon dioxide come from? From the air. Okay, this is how plants use carbon dioxide from the atmosphere for photosynthesis. Now, sticking atmospheric carbon dioxide onto RUBP is not something that just happens naturally very quickly. So as usual, we're going to need an enzyme to speed up this reaction. And the enzyme is represented by this little purple box. It's called Rubisco, a shorthand. It's got a longer name that ends in A's, but we're not going to worry about that. We'll call it Rubisco because that's shorter and easier to remember. So when you join the 5-carbon RUBP with a 1-carbon carbon dioxide, we're getting this 6-carbon molecule, which you don't have to know the name of. But you do know that there are 3 of them, right? 3 RUBPs, and each one of those gets a carbon attached to it. It makes 3 6-carbon molecules. Still have 2 phosphates on the end, those 2 yellow dots. Now this isn't a very stable molecule, so it breaks in half. So the 6-carbon molecule now becomes 2 3-carbon molecules. So we originally had 3 molecules of that unstable thing. Now we've got 6 because each of those divided in half. Hopefully you're with me so far. This little burst of energy should look familiar. Hopefully remember that's ATP. We need ATP for our next step. Where do we get that ATP from? Oh yeah, we made ATP back in the light reactions. Okay, so this is one of the areas where that ATP from the light reactions that's made in the electron transport chain is needed. So we turn ATP into ADP, which means you're going from a triphosphate to a diphosphate. What do we do with those phosphates? Stick them on the end of this new molecule. See how we have 2 yellows on the end, where before up here we only had 1 yellow on the end. So we use the phosphate from ATP and add it onto that 3-carbon molecule. And since we have 6 of them, we need 6 ATP. So to go from these 6 molecules to these 6 molecules, we need 6 ATP to turn into 6 ADP. All right, the next step is going to be the other thing we made in the light reactions, the NADPH coming in. And remember, just like I said NADH back in cellular respiration, its whole job was to carry hydrogens around. Same thing here. Okay, this is where it uses its hydrogen. It's going to drop that hydrogen off. And now it's going to be NADP. That hydrogen and the electrons associated with it are used to kind of rearrange this 3-carbon molecule right here and change it into a different 3-carbon molecule. Again, you don't need to know the name of it. And there's still 6 of them. All right. Once we do all the rearranging, this final molecule that's been made is called G3P. Now that should sound familiar. G3P was what we made back in glycolysis when you took the glucose and you split it in half. Each half of a glucose is G3P. So now we've got the puzzle pieces needed to make a glucose. And actually G3P can be used to make lots of different carbohydrates that the plant might want to make. Remember, there's all sorts of carbohydrates like starch and cellulose and other things that we didn't talk about that plants might want to make from G3P. So we've got 6 G3P that have been made. One of them, so 1 G3P, actually is available to use. All right, so out of those 6, 1 G3B goes and potentially can be made to make glucose eventually when we've got enough G3P stockpiled. However, the other 5 G3P need to go on and be recycled so that the Kelvin cycle can continue. So out of this original 6 G3P, 1 gets used to potentially build a carbohydrate, and 5 get passed along through the cycle. That's why I wanted to start with 3 carbon dioxides and 3 RUBPs in the beginning, so that you could see how they get recycled. So we've got 5 G3Ps here. Okay. Remember that each of these contains 3 carbons, right? Which means we have a total of 15 carbons to deal with right now. RUBP is a 5 carbon molecule, remember him up here? So that means with our 15 carbons from G3P, we can rearrange them to make 3 5 carbons, so 3 times 5 is 15, so we can rearrange them to make 3 5 carbon RUBPs. But that rearranging takes some energy. Here's our energy again. Again, if we're making 3 RUBPs, that means we're going to be using 3 ATPs, turning them into 3 ADPs, so that we can rearrange those G3Ps to make RUBP. Lots of P's, lots of phosphates involved in this. And then the cycle starts all over again. More carbon dioxide can bond with those 3 RUBPs, and we make some more G3P. Okay, this looks like a mess. So that's why we're going to move on and draw this again so you can have this in your notes from scratch, and it's also a good review for you. So let's do your diagram for your notes next.